I'll make my life easier. Okay, so digestion. So we are going to talk about two main things. The real big thing I want you to get from digestion is I want you to be able to know the secretions and the hormones that control said secretions. So that's what we're going to focus on for these two PowerPoint slides. If you can get further in the weeds than that, great. But if you at least know this, you will be pretty well set up for a lot of your physiology questions of the digestive tract. All right, some of this stuff we're going to fly through. So enzymes are secreted initially as zymogens. So all digestive enzymes are secreted as inactive forms, which are called either zymogens or proenzymes. You'll see both of them thrown around. You have to undergo then proteolytic cleavage to activate the next set of enzymes. So they have to be cleaved, they have to be essentially activated after they're secreted in their inactive state. Why? This is protection, right? Our digestive enzymes digest stuff. If we have them secreted and we have nothing to digest, what are they going to digest? Your tissue. So we keep them in this inactive form, especially if they're secreted during times where we're not digesting, to protect our own body. It's like a self-protection. You can think of it like tolerance as involved in the T cell system. Prior to secretion, zymogens will be in granules by the cell membrane, ready to go. And like these vesicles, much like a neurotransmitter, is sitting in the synaptic terminus in a vesicle waiting to be released. The granules, like the terminal or the vesicles, will fuse with the cell membrane and expel their contents into the intestinal lumen. So when I'm talking about secretions of the digestive system, I'm talking about things that get pushed into the lumen, things that get pushed into these digestive spaces. When I talk about hormones, remember, those are going to go to the blood. So that's different. So the key piece will be recognizing if it's a hormone or recognizing if it's a secretion. If it's a secretion, it's going into the lumen of whatever space. If it's a hormone, we're going into the blood, and it can affect the body throughout the whole body. So some important gastric and pancreatic zymogens. So in the stomach, these are the site of synthesis. So stomach versus pancreas. We have the zymogen listed here, the active enzyme that it becomes, and whatever activates it. This is a great place to start for some of these enzymes, these important enzymes. Pepsinogen becomes pepsin, and it self-activates when pH is low. Pepsin also can convert pepsinogen to more pepsin. So it has this positive feedback cycle, which is set within a greater negative feedback loop. But this could be a localized, a small example of positive feedback, okay? When the pH gets too low, or if the pH changes and is not in its ideal environment, that's going to not let pepsinogen become pepsin. You also will see that other hormones and things will see, oh, the pH is low. We want to suppress the pH from getting lower. So that's the negative feedback loop that kind of helps control this process for pepsin. Chymotrypsin, trypsin, carboxypeptidase, and elastase. All of these enzymes, we can see their zymogens here, and we can see that the majority of them, chymotrypsin, carboxypeptidase, and elastase, are activated when trypsin is present. So really, we need trypsinogen to become trypsin for us to be able to activate our other digestive enzymes. If this process occurs, it's like a cascade. The rest will occur. And so what is trypsin's activator or trypsinogen's activator? It's enteropeptidase, which this is located on small intestine epithelial cells. Entero meaning small intestine or gut, and then peptidase, it's going to help digest proteins. And so when this is present and this is getting activated or released, it's going to trigger a trypsinogen to convert to trypsin. And that's how this process can take place. If trypsin's present in the lumen, the rest of these enzymes, when they're released, they'll be triggered to be released, and then they will become their active form. Now, the exception to all of these is alpha amylase, which is secreted by saliva and pancreas, is secreted in its active form. So alpha amylase is the one that's secreted active. It does not need to be converted into its active form. And alpha amylase digests what? Carbs, carbohydrates. Yep, good. So we start with chewing and then we'll swallow. Saliva is important. Not only does it contain alpha amylase that helps cleave 1,4-glycosidic bonds. So these are bonds that are in a lot of our carbohydrates. So this is how it's starting that process. So our chewing, our saliva, creating the bolus, the manual part of digestion. We have several manual parts of digestion. The alpha amylase, the actual chemical portion of the digestion, all happening in the mouth. Polysaccharide breakdown in the mouth is limited. We're not necessarily getting to disaccharides fully in this form. We are just breaking them from polysaccharides to smaller polysaccharides. Protein digestion is going to really not even begin outside of mastication until we get into the stomach. Stomach acid and pepsins is really what starts the majority of protein digestion outside of the manual mastication, which is just breaking down food. But the acid and these pepsidases or these pepsins they denature the protein, but we need the stomach to be in an acidic environment. The hydrogen bonds are able to be broken because of low pH. So that's essentially what we're doing is we throw a bunch of acid into the system, we nature a bunch of enzymes, right? If you ever played in chemistry or OChem, you had enzymes or proteins, you add some hydrochloric acid, there's color changes like our titration spectrums, like whoever had to do titrations, you probably all did. So all these different pieces of the puzzle you see activated or as an example of in our stomach with this really acidic environment with hydrochloric acid. Then our proteolytic enzyme pepsin also will start working on proteins in the stomach, can break them down into smaller fragments. We start to get our proteins broken down into smaller peptides, but our goal is to get them to be individual amino acids because that's what we can absorb and use. On stomach acid, the gastric proton pump, the hydrogen, potassium, ATPase, meaning it's exchanging hydrogen and potassium, and it's using energy to do its job. It's located on the cells lining the stomach. It is very much like the sodium potassium ATPase pumps we've discussed previously, but its job is to pump protons, hydrogen ions into the stomach, and it takes out potassium and it uses energy. So this process of creating an acidic environment isn't without cost. We are taking away potassium, so we need some potassium in the actual area, in that luminal space to exchange for our hydrogen. And then we're going to, we also need energy. So if we're in a fasting state, that could be harder, or an energy depleted state, a malnourished state, would be more challenging. Once we move to the small intestine, where our partially digested proteins, carbs, and lipids are going to move through our pyloric sphincter, the acidic chyme or the low pH mixture in the small intestine is going to stimulate our small intestinal cells to release secretin, which is a hormone. So now we have our first hormone that we've discussed in this one. We'll talk about some other hormones in our next slide set. But secretin is released because of that transfer of acidic chyme into the small intestine. That's going to tell the small intestine, we need to protect ourselves, so you need to secrete bicarb. So secretin's one of its main jobs is to help secrete sodium bicarb from our pancreas to neutralize the acidity that just got passed through our stomach. From there, the polypeptide products of pepsin, try to say that 3 times fast, stimulate, so seeing those polypeptides, stimulate the release of cholecystokinin, which is also a hormone, also released by intestinal cells. The pancreas will respond to CCK by releasing its kind of cascade of digestive enzymes. Those enzymes held in their inactive forms in vesicles will now be stimulated by the hormone to be released. And they will be released and they will move down the pancreatic duct through the ampulla vator into the small intestine, just like the anatomy we learned and relearned yesterday. That will help with starting digestion or continuing digestion of lipids and finishing or almost finishing the digestion of carbohydrates as well as protein. So this is like our little picture here. It's a very kind of small biochemistry view. You can see food going in here, proteins plus hydrochloric acid plus pepsin. We get to oligopeptides in our stomach and initial digestion products from just kind of the churning of the stomach itself. Once we move over through that pylorus region, the pyloric sphincter, we're going to have CCK and secretin. They'll be activated by those oligopeptides and by those initial digestion products, the acidity as well, to tell the pancreas to secrete bicarb as well as its digestive enzymes through the ampulla of Vater. And then we can finish up our digestion process. Okay. The final job of protein digestion, the pancreatic proteases will break down proteins into oligopeptides. So again, making them smaller and smaller. And peptidases will finish the job of protein digestion, which is they're attached to the external surfaces of the intestinal cells, our brush border enzymes. That's our peptidases. So anything that affects our brush border, we could think of multiple conditions, could potentially affect this last stage step in a lot of our digestive processes, but specifically for proteins. making it challenging for us to break our oligoproteins or oligopeptides down into individual amino acids for absorption. But they will cleave these oligopeptides into amino acids or di or tripeptides, and then they can be moved into the actual cells of the small intestine. And there's 7 types, and they're specific for specific amino acid groups. You do not need to memorize the 7 types of transporters. In fact, do not do that for this exam or ever. Then once those amino acids have been absorbed through the cell, then they'll be released into the bloodstream, so then the body can use them. Okay. Gluten or no gluten, internal question we always get. So celiac disease, there's many, it's multifactorial, but there is a component of celiac disease, a big component that's caused by the inability to digest certain proteins. Celiac disease, we see it as a gluten enteropathy. We'll look a little bit more at some of these digestive pathologies coming up next week, but we will see intestinal inflammation triggered by proteins in wheat, rye, and barley. And then gluten itself, the protein gliadin and glutenin in wheat and secalin in barley and rye are specifically some of the proteins that the body has a response to. All of these proteins have a lot of proline and glutamine and amino acids in their structure. And so our body will see complete digestion resistance by pepsin and pancreatic proteases, which then leads to inflammation. It's a genetic component there that can damage our intestinal epithelial cells, which affects our absorption of other stuff. So it's this big inability to absorb cascade. Over time, that inflammation causes these villi to become blunted, decreasing surface area and further affecting our brush border enzymes. That's how you can kind of see the pathogenesis of celiac disease occur. This stat is old, it's a 2022 stat. So I would probably want to update it. 1 in 250 people have this condition. I also don't fully trust that stat. Carbohydrate digestion, alpha amylase and more. So we talked about carbohydrates kind of getting into the stomach using alpha amylase initially in saliva. Once it gets into the stomach, our stomach is doing that churning process, but it doesn't hit amylase again until it gets to the pancreas and the small intestine. Carbohydrates, the goal is to get them down to disaccharides and monosaccharides, glucose, fructose, and galactose, so that we can absorb them and use them in the body. Alpha amylase cleaves alpha 1,4 bonds, but doesn't cleave alpha 1,6 bonds. So you can get to di and trisaccharides. And then both maltose and maltotriose and limit dextrin are essentially the sugars that are not digestible by alpha amylase because they contain alpha 1,6 bonds. And so these then need an additional enzyme. So alpha glucosidase digests the rest, except for dextrin, which is broken down by alpha dextrinase. Both are brush border enzymes. Okay. How much of this do you need to know? I've seen, unfortunately, I've seen questions on both of these enzymes before. I would say alpha amylase, you already know it, so you don't technically need to relearn that one. So maybe learn alpha glucosidase, and then if that one feels confident, then alpha dextrinase. But know that they're both brush border enzymes. And then if you're like, what the heck am I talking about? Alpha 1,4 versus alpha 1,6 bonds. We can see the 1,4 bonds are able to be broken, but these pink 1,6 bonds, those are the ones that alpha amylase cannot break. And that's why you need these other enzymes to break them down. Alpha amylase is not a brush border enzyme, correct? Alpha amylase, we'll see in the, you'll see a salivary amylase and a pancreatic amylase. Awesome. Your disaccharides are going to be easier for you to digest than obviously complex carbs. So sucrose and lactose. Sucrose is glucose plus fructose. Lactose is glucose plus galactose. Sucrose in veggies digested by sucrase. Lactose in milk digested by lactase. Both of them are also on surfaces of intestinal cells. So another brush border enzyme. Glucose and galactose, then you can transport directly into your epithelial cells. They're transported by sodium glucose linked transporters, SGLTs. These you can see are co-transporters of glucose and galactose, bringing in both the sugar and sodium into the cell. The sodium is the one that's driving this transport because the sodium is moving down its concentration gradient. from extracellularly to intracellularly. So that's what helps drive this co-transport process. Fructose gets its own transporter, GLUT5. So fructose goes through its own special tube because she's different and needs her own place to be. And then all of them can be transported through the GLUT2 receptor into the blood. Okay. Lipids are more complicated for digestion. Proteins and carbs are easier. It's because they're highly reduced. So their hydrophobic process makes them a little harder to break down. And they also have a lot of energy. So we want to break them down. When we digest lipids, we kind of start in the stomach. I mean, we do mastication. And then in the stomach, we kind of get to start doing things with the churning. We ingest them in the triacylglycerol form. And we need to get them to fatty acid form so that they can be absorbed. So that's our goal, is from triacylglycerols to fatty acids. Lipids are prepped to be digested in the stomach, and that's through the churning, the grinding and mixing that makes an emulsion. So if you think about that, think about your vinegar and oil in a salad dressing, right? When you add oil to your vinegar mixture to make dressing, if you just combine them together without mixing them at all, they'll stay separate. But if you shake or you stir or you try to emulsify it, it's the same thing that the stomach's doing. It's emulsifying your fat, your hydrophobic stuff with your hydrophilic stuff. So the same thing is happening. You're just making a big nasty salad dressing in your stomach. Your vinegar is your HCl, and your whatever you ate as fat is your oil. Once the lipids move to your small intestine, bile salts help with that emulsification. And so this is where the gallbladder and bile comes into play, and it has its really big job. Bile salts are formed from cholesterol made in the liver, secreted by the gallbladder when cholecystokinin, CCK, is present. So that hormone, again, also will affect the gallbladder secretion of bile. So hormones, super important. Bile salts bind to lipid droplets, and they make a change in the product. Once those bile salts are bound, it's essentially going to make the bond weaker, so it's more likely to be able to be broken. Then we have lipases. We have bile salts that bind to them, and they make them more soluble. Then we have our lipases, which is a pancreatic enzyme that will break down triacylglycerols to free fatty acids and monoacylglycerol. And then the final products of lipid digestion are these two products themselves. They're carried in micelles, essentially carried away so that the fatty components are tucked away from the watery components of the digestive tract to the plasma membrane where they are absorbed. These are globular and they're formed by small lipids. The polar heads are outward, the fatty tails are inward. Bile salts themselves help to stimulate micelle formation. So in someone who has inappropriate bile secretion, or who has a gallbladder that's removed, and maybe their bile storage and their bile secretion is either dripping, secreting too much, or blocked or not secreting enough, we can see issues with fat digestion, which is why you can get steatorrhea, fatty stool that's very foul smelling and oily, because your body can't actually absorb the fat because it's not able to break it down and it's not able to transport it. If you want to see the full meal deal process, we got our lumen, our blood, and our cell, triacylglycerols, add some water, some lipases, get our fatty acids and our monoacylglycerols, transport that via our micelles through this fatty acid transport proteins, FATP or FABP, depending on the book that you use. FABP is the binding protein and TP is the transport protein. So it's technically both. So they go bind first and then they transport next. Then triacylglycerols are actually resynthesized from free fatty acids and monoglycerol in the cell itself. So we have to break it down to transport it to reform the thing. So our body has to do a lot of work with fats, but they're a big energy source. So it's worth it for our body. The resynthesized lipids are then associated with a protein, and a small amount of phospholipid and cholesterol are formed in this actual lumen of the smooth endoplasmic reticulum. And then that's where your chylomicrons are formed, which they're transported, all the newly formed molecules of your fats to the external tissue and system. As we saw with our test question Tuesday, chylomicrons were the thing that transported our digestive triglycerides to the body. And then whichever ones aren't used or stored, it's going to go to the liver, those other products. Cool. Digestion. We've done it. Yes. Oh, yes, sir. Yes. Yeah. Yeah, it goes into the lymphatic system because it's easier for us to transport fat through that system. It has a little bit more room, a little less crowding than blood does itself. So it's a little bit of a faster highway than the venous system or even the circulatory system, the blood system. How it gets there, they're in the lymph nodes and the spleen. That's where the communication system happens. So most of the time it's going to stay in the lymphatics until it gets to a lymph node or until it gets to a lymph organ of sorts. So maybe the thymus, not really, or the spleen. That's where you'll see essentially stuff will exit out of the lymphatics highway and then can exit into the circulatory system. And so that's where that exchange happens. Partially why the gut has so many lymph nodes. And it is kind of a lymphatic organ in itself, is because you could even do that transport system right there locally in the gut. And the liver itself produces about 25 to 50% of our lymph fluid in the body. And so our liver is actually a big lymphatic organ. We just don't talk about it that much. And so a lot of that exchange can happen right there, pretty locally, without having to go through all of systemic circulation, versus if we went through the blood, we might not get to the liver quite as so fast. We would probably have to go all the way through like the cardiovascular and back out, which would take more time. I was literally just talking about the lymphatic system to a preceptor yesterday and how we never really discuss it, but it's one of the key systems we modulate and modify as naturopaths. So yeah, I think it's great, and that's why it's on here. We just kind of skip over it. It's like, well, it happens, but it's key. It's important. Yeah. Does that help kind of explain the why? Okay, great. Thank you. That will be in the liver. Yep. Lymph into the blood, it'll take into the tissue. So any triglycerides that need to go to certain tissues for energy or for storage, that will happen. And then the remnants will go into the liver, where it's then transported via VLDL, and you have that VLDL, IDL, LDL, HDL breakdown and formation. Does that help? It has to be packaged and transported first, and at the liver, that's where all these good things are formed, good and not good, depending on who you talk to. Yes. Did you have a hand? Okay. Correct. It will travel via its chylomicron into lymph first, and then it can go into liver. Yeah. And we will go through cholesterol synthesis pathway when we do biochem. So that'll be one of the pathways we focus on. So we will get that whole process of formation of the LDL, the IDL, HDL, all of it. It's asked about enough, and I feel like we forget about it enough that it's worth revisiting. Awesome. All right, I say we break, and then what we're going to do is we'll finish regulatory of the gut. We'll do some kidney, and then if we need to push some cardio to next week, we have the time, so we can. All right, so let's do a little shorter break, a 7-minute break this time, and then we'll start back at 11:10.